Oh, for goodness sake. Okay. Okay, good afternoon, everybody, and welcome back. Remember, when you join, you are automatically muted, but you can unmute yourself at any time to ask a question. And so, welcome to you all, although I wouldn't say that I can see you in the same way that I would like to be able to see you in class. Nonetheless, it's nice to know that you're out there. Okay, so we've been talking about bacterial growth. Now, normally when I teach this class, I don't go into as much detail as I'm going to go into in this class, but I'm going to cover some aspects of bacterial growth, such as culture techniques and things like that, that I don't usually cover because I know that you have a restricted number of laboratory sessions. And these are things which we would normally cover in some detail when we did a lab on culturing bacteria, etc. So it's useful for you to at least hear from me now. Let's remind ourselves of where we left off last time, talking about one of the most important of all of the culture conditions that we observe when we're growing bacteria. And that is the importance of the oxygen in the environment of the bacteria. And I'm just quickly going to run through this again. We recognized prokaryotes that were obligate aerobes. That is, they are required to have oxygen present in some abundance in order for them to function properly. They have no ability to function anaerobically, so they are obligate aerobes. We see them in what you'll learn in a little while is called a reducing medium. We see them only growing here at the very top of the tube where oxygen is abundant. Very, very frequently, you will encounter facultative anaerobes. Now, facultative anaerobes are organisms which grow in the presence or the absence of oxygen. And they are able to switch. They are able to switch from cellular respiration to just using glycolysis and fermentation in order to survive. So when we grow them in a tube like this, we'll see they grow throughout the tube, but they grow best at the top because aerobic growth is much more efficient. Metabolically, it's much more efficient. It produces far more ATP than glycolysis alone. So they will grow, and that's very useful for them because it allows them to penetrate into anaerobic habitats as well as occupy aerobic habitats. There are some prokaryotes which are obligate anaerobes. They positively require the absence of oxygen. They are totally intolerant of the presence of oxygen. In a tube like this, they will only grow at the very bottom where it's absolutely, completely anaerobic. These organisms are often extremely fastidious about oxygen. Even a few atoms of oxygen in their environment, they will not grow. So they are obligate anaerobes. These ones here we didn't talk much about. They're not of great importance for our purposes. These are called aerotolerant anaerobes. They grow anaerobically, but they tolerate the presence of oxygen. They just don't use it. But we didn't spend much time talking about them. And lastly, a group called the microaerophiles. And microaerophiles will grow in the presence of oxygen, but they don't like an abundance of oxygen. And they don't like total absence either. They like some oxygen, but only a small amount. And these are important because some clinical pathogens, for example, are microaerophiles. And that stands to reason. If you think about where pathogens grow in our bodies, for example, they will very frequently be growing in areas where there's not an abundance of oxygen, but there may be some oxygen present. And an obligate anaerobe would not survive, but a microaerophile can. So those that we covered last time, and it's really important. Make sure that you understand that. Okay, so we're going to be talking today mostly about how bacteria grow and about how the processes of growth take place, and then also a little bit about how we manipulate that in the laboratory. The first thing to understand, very, very often, bacteria grow best when they're in contact with a surface and when they have some contact with other bacteria, especially of their own species. And for that reason, surfaces are extremely important to bacteria, and they will very frequently form a community on a surface. And that we refer to as a biofilm. It has a particular structure. It's not just that the bacteria happen to land there. They're actually often there is a requirement for them to adhere, for them to grow properly. And that is one of the functions of that exterior material that we saw before, all that material that is outside the cell wall, things like the capsule and the slime layer. These things are often a mixture of substances, often complex polysaccharides of one sort or another, which provide an adherence capability, so they can stick to surfaces and they can stick to one another. And that is indeed what they do. It's an amazing thing that bacteria actually can communicate with one another over a distance. And bacteria will often not grow properly until there is a sufficient number of them present and metabolically active. This is called quorum sensing. You know, the quorum is like in a meeting where you need a certain number of people present in order for the meeting to begin. Bacteria do the same thing. They emit signaling molecules which can be picked up by other bacteria. And only when they begin to pick up sufficient of these signaling molecules called inducers, only then do they begin to grow properly. And in growing properly, they begin to attract even more bacteria to where they are. This provides all sorts of benefits for them. The capsules and slime layers, whatever, start to build up one on the other. They stick the bacteria firmly to the surface. And bacteria may begin to set up what we refer to as a consortium. And a consortium is several different species of bacteria that are cooperating in the film, and they begin to share nutrients. One bacteria may produce excess of a nutrient which the other bacteria can use. It is a cooperative system with huge advantages, not least of which is that it's actually quite difficult to get that biofilm off. It adheres quite tightly to the surface, so the bacteria have a safe environment. That biofilm also may shelter the bacteria from all sorts of environmental features. And one of the really important ones is that our own microbiota very often forms a biofilm. We have a biofilm on our teeth, for example, on mucous membranes. We have biofilms in our gut. And that's a normal kind of situation, non-pathogenic, but they can also form films which are of pathogenic bacteria. And the biofilm actually protects them from our immune system, for example. It restricts the ability of the immune system to react to the presence of the pathogen. And we're going to hear later on this can be an extremely dangerous situation. And bacteria may be specially adapted to developing a very vigorous biofilm, which the immune system finds very difficult to penetrate. The biofilm has a particular physical structure. It's not just a thick layer of slime with bacteria embedded in it. Instead, it usually is formed into they are kind of pillars, clumps of bacteria which grow vertically more than they grow horizontally. And this allows percolation of the aqueous medium, whatever it is, whether it's, if it's a natural biofilm, this would just be the water around it. If it's a biofilm which is forming inside the body, then it would be body fluids around the biofilm. But it allows the movement of oxygenated water, for example, oxygenated aqueous solution, to move through, carry away waste, bring nutrients, etc., etc. This is a very, very advantageous system for bacteria to grow in. And many of them will not grow properly if they are prevented from forming a biofilm like this. In addition, notice that the biofilm can reproduce itself. This clump, for example, may be very complex. It may be several kinds of bacteria at different stages of growth doing various things and cooperating with one another. That these clumps can break off and then go and form another film somewhere else. They can settle elsewhere and start a new portion of the biofilm. this is a very common feature of bacterial growth under natural circumstances. In clinical settings, it's extremely important because these biofilms form very easily on artificial surfaces. And for example, we have lots of natural ones, of course, but if we look at artificial surfaces, pipes, tubes, catheters, all of these kinds of situations, biofilms will very rapidly form on the surfaces of those items. And a big problem is the structure of the biofilm, the capsular materials around the bacteria often make them resistant to disinfection. And it won't surprise you to learn that a lot of hospital-related, medical-related infections are related to things like catheters, heart valves, contact lenses. All of these sort of things are excellent surfaces for bacteria to grow. And when you have that kind of situation, you don't have to have growth necessarily of pathogenic bacteria, although that can definitely happen. But even innocuous, normally innocuous bacteria growing in these circumstances can prove to be extremely dangerous. And they can be very difficult to get rid of. It can be very difficult to manage these catheters, heart valves, these sort of things, once those kind of infections are set up. The one which biofilm, which when it goes wrong, affects almost all of us to one extent or another, is the biofilm on our teeth. And this is under normal circumstances, we have a healthy microbiota inside our mouth, which is well balanced. There are lots of species involved in it. And the biofilm that forms naturally on our teeth causes no problems. But it is very, very subject to vagaries of diet. And so if we begin to feed that biofilm with the wrong kinds of nutrients, the nature of the biofilm changes, the species composition changes, and we begin to get bacteria growing which are in fact pathogenic. And the most important, for example, here's a biofilm. This is actually onto a plastic surface, but this could as easily be a picture of a dental biofilm. And tightly adherent to the surface, this is all capsule material, tying it tightly and toughly to that surface. It's difficult to get rid of. In the case of dental caries, dental decay, we start to get species, especially which are fermentative and start to produce acids. So say this was a tooth surface, they're releasing acids around them, and the acid begins to decay the enamel of the tooth. In addition, this capsular material, in the case of teeth, of the tooth biofilm, begins to accumulate mineral deposits, and it forms a hard layer which becomes irritant to the gums, for example, and that is dental plaque. So this would constitute dental plaque if it had the pathogenic qualities which would lead to both tooth decay and to gum disease. So there's a very important biofilm, and the cause, by the way, of billions of dollars worth of lost time and a tremendous amount of grief to people who suffer from tooth decay. Okay, so we're going to talk a little bit about how we actually grow bacteria, especially in the laboratory. I first of all mention that in the United States, all laboratories dealing with biological materials, for example, or any scientific materials, are rated. So we, for example, in our laboratories, in our microbiology and biology laboratories are inspected, and we're assigned a rating. depending on what we're planning to do in those labs. So a BSL-1 lab, this would be basically your ordinary general biology lab, where you're not, if you're dealing with anything, you're probably dealing with preserved specimens, that sort of thing. Unlikely to be dealing with anything which even had a remote possibility of being pathogenic or dangerous. BSL-2 lab, this is the kind of lab, this is your microbiology lab that you would use at St. Francis. It's a BSL-2 lab. We are permitted to use live bacterial cultures, for example, but we're only permitted to use live bacterial cultures which have a low pathogenicity. So the likelihood of them causing any disease, even if you were to be contaminated, is low. Nonetheless, in a BSL-2 lab, you are required to wear a lab coat, gloves, eye protection whenever. This eye protection is a somewhat loose rule. We usually recommend eye protection where there is any danger of splash occurring. So this is really quite minimal precautions. In addition to that, the behavior of people in the lab is stressed. You stress disinfection of bench tops, for example, before and after use, disinfection of the hands before and after a lab, and all of these kind of common sense precautions. A BSL-3 lab is one which you won't encounter at a college like St. Francis, but a BSL-3 lab is one which deals with organisms, for example, which are overtly pathogenic. And maybe it's clinical microbiologists studying pathogenic bacteria, whatever. And those kinds of cultures, let's say you were working with anthrax or tuberculosis or something like that, you would only be using those bacteria, only exposing them inside an enclosed biosafety cabinet, along with numerous other precautions as well in the lab. But it's an observation that you are dealing with potentially pathogenic organisms. BSL-4 is for dangerous, highly infectious organisms. So, for example, if somebody is dealing with COVID-19, for example, which is properly called SARS-CoV-2, then they would be working in a BSL-4 lab. And this is a lab with negative pressure, air pressure inside. That is to ensure that everything stays inside that lab. Anything that's liberated in that lab stays inside the lab. It does not get out at all. A BSL-4 lab very usually will have showers to go in, showers to go out, and extensive precautions, often complete covering with personal protective equipment and that sort of thing in order to do the work. All air leaving the facility is filtered, and it's double filtered through special filters which will take out even viral size particles and ensure that nothing is disseminated. So if you were to be in a lab at St. Francis, you would be in a BSL-2 lab. Okay, so how do we do the work in a lab when we're dealing with bacteria? How do we actually grow these things, and how do we know what we're dealing with? Well, the first thing, there was some terminology. The first is, you will hear me often refer to culture media. In a culture medium, there are many, 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 many types. But a culture medium is a preparation of nutrients which are suitable for growing the bacteria that you're interested in. In our case, many, many of the bacteria that you might encounter in the lab are pretty generalist. We would be able to grow them on a pretty general kind of generic media, as you'll see in a minute. But sometimes bacteria require special media. They require carefully made media, which is particular to their particular circumstance, to their particular metabolism. Many bacteria are extremely difficult to grow, and the development of media, culture media for those bacteria is a whole science in itself. The media that we use is always sterilized. It is sterilized to ensure that we have control over what grows on it. This is a big problem in any microbiology lab, and that is that we want to keep our cultures pure. We want to prevent contamination. And it should explain that bacteria don't fly around in the air. They don't move on their own in the air. In general, contaminants like bacteria, fungi, these sort of things, which might contaminate culture media, usually come in the form of attachments to small dust particles. The most important, the most abundant source of contamination in a lab is actually the workers themselves, because we produce skin flakes the whole time. It's part of our natural skin structure. Those skin flakes carry bacteria on them, and that is the most frequent source of contamination on culture media. Before we even start, we need to ensure that the media we're using is sterile. And that usually means that we have put it through an autoclave. That is a pressure cooker in which we are able to raise the temperature well above boiling point. And that ensures that we kill not only living bacteria, but that we kill bacterial spores as well, which is another major source of contamination. If we simply boil media, we may actually still have bacterial spores in it, and those will revive once we reduce the temperature below boiling point. In order to kill spores, we have to raise the temperature well above boiling point and keep it there for about 20 minutes and in an autoclave, and that ensures that everything is killed and that the medium is sterile. Once we have a sterile medium of whatever kind, we can inoculate it. An inoculum is a sampling of bacteria of known identity. So we take a sterile medium, we inoculate it with known bacteria, and we can then be fairly sure, not be absolutely sure, but we can be fairly sure that whatever grows in that medium is going to be of the same identity as our inoculum. That growth we call a culture. So we're going to look at we're going to make our culture medium, we're going to sterilize it, we will inoculate it, and we'll culture it and see what happens. If we were in the lab, most of our cultures will grow very well at 37 degrees Celsius. Most of that bacteria, in fact, will grow pretty well anywhere from room temperature through 37. Why? Think back what we talked about last time, because most of the bacteria we're dealing with are mesophiles. So they have an optimum around 37. It can vary a bit either side, but they grow well in that range. Room temperature 37 is usually ideal for most of the things that we would deal with in a lab. Okay, so as far as the culture media are concerned, the first thing is that there are two groups of media. The first kind of medium is one where we know exactly what is in the medium. For each component, we have weighed it out. We can describe it either by weight or by molarity or something like that. We know exactly for each component exactly what's there. Chemically defined media, that's what they're called, are very often needed where we grow those difficult organisms, the fastidious organisms. And the reason for that is what makes those organisms fastidious is that they often require the addition of growth factors that more generalist bacteria do not need. And there are many, many, many recipes for chemically defined media for growing specific bacteria. In our laboratory settings, we would seldom use them. In most clinical labs, the chemically defined media are confined to studies of very, very specialist kind of bacteria. Much more commonly, what we would do would be to use complex media. And complex media, really, we know what's in them, obviously, because we make them. But we can't say specifically. We can't tell you, well, I can't tell you there is this molarity of glycine, an amino acid. I can't tell you there's this molarity of vitamin B, etc. All I can do is tell you there's plenty of amino acids, there's lots of vitamins of all sorts, and there's plenty of stuff. There's abundant carbon, abundant nitrogen, etc. And we'll see how we build this up in a minute. There are certain standard things. These have been used literally for centuries in these media. The first is we often include digestive yeast or something like that. That digestive yeast, it provides all sorts of things. It does provide amino acids, nitrogen, carbon, all sorts of things. But very importantly, it provides vitamins, growth factors, which are maybe necessary for the bacteria we're growing, but are certainly helpful to them. Then we often include a digest of proteins. That protein can be from meat, sometimes from plants, like soy protein is often used. And all that happens is that they will take that source of protein and enzymatically digest it. And often the name gives you an idea about how it was prepared. You will often see, for example, that we include peptone. Or peptone, all that peptone is protein that has been digested with pepsin, which is the enzyme which we produce in our stomach. Tryptone is another one. It's also a protein mix from meat or whatever, which is digested with trypsin, which is a pancreatic enzyme, which digests protein. So all that enzyme does takes the long protein molecules and it chops them up into smaller pieces, chops them into little tiny oligopeptides or little tiny peptide chains and single amino acids. We can't actually say exactly what is in them, but we know it's a good rich mixture of amino acids first of all, but also as a source of carbon and of nitrogen as well, of all sorts of things that the bacteria can use. We may sometimes be required to add a couple of others. Some bacterial media sometimes, for example, call for a little bit of extra phosphate to be put into them as a phosphate salt or something like that. But these complex media are basically aqueous media, which are a solution of at least yeast extract, a protein digest like tryptone and peptone, and that would be about it. If I just take water and I dissolve yeast extract and then peptone or tryptone in it, it's a very good growth medium for many, 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 many bacteria. And that I would refer to as a nutrient broth. And soup, broth is a soup, and that's literally what it is. It's a nutrient soup for the bacteria to grow in. Nutrient broths are frequently used. You would, in lab, would often use them for general growth. And they're very, very good. You can grow bacteria rapidly in them. Most bacteria grow very easily, but there's one big disadvantage to using a broth. And that is, you can't really tell what's growing in your tube. One bacterium looks very much like the other when it's growing in a nutrient broth. It just takes a clear broth and it makes it go turbid, it makes it go cloudy. And if you've inoculated with a particular bacterium, you want to be really sure that what is growing is what you put in. It's not some contaminant that dropped in while you were doing it or something like that. It's difficult to tell with a nutrient broth. And there's various other disadvantages. It's sometimes difficult to tell how many you have in a nutrient broth. It's very advantageous in microbiology to have a solid surface. And I told you when we talked about history that this was actually, as simple as it is, this was a major breakthrough in microbiology, was the development of a suitable solid medium. And basically a solid medium is a nutrient broth. It's all the nutrients we have in an aqueous solution, solidified with something so that it forms a gel, a tough solid gel that where we can work on the surface of the medium. And that we usually use agar. And agar is an extract from seaweed. And it has certain advantages, which I'll describe in a minute. But one of the huge advantages is that there are not a lot of bacteria which will digest agar. You can also solidify nutrients broth with gelatin, for example, which is an animal extract. It's jello. But the problem with that is that many, many, many bacteria happily digest gelatin. And so you can make up solid plates, inoculate it with bacteria, come in the next morning, and they've turned the whole thing back into a nutrient soup, into a nutrient broth, because they digested all of the gelatin. That doesn't happen usually with agar, because bacteria in general don't have the enzymes to break down agar. Okay, let me just stop. Are there any questions from anyone? How many of you? Yes. Hello. Anyone? All right. Don't forget, unmute and yell at me if you need me to slow down, repeat, or answer a question. Don't feel shy about interrupting. Okay, so this agar is very, very important. And if you stay in microbiology, and especially if you move on to molecular biology and other studies, you'll find that agar is a substance which makes its appearance over and over and over again because it is such a good gelling agent. It's polysaccharide, but most bacteria that we would deal with will not digest it. The one place where it becomes a problem is if you're dealing with marine bacteria. Think about that for a minute. That marine bacteria quite often do digest agar. Why would that be? Well, look, it's a complex polysaccharide and it's derived from seaweed. So marine bacteria might encounter it and have developed the enzymes to digest it. The kinds of bacteria you encounter in a clinical lab, they've never seen it. They're never in their evolution have they seen agar, so they don't respond to it. So to make agar medium, we take a nutrient broth, we put agar in it, and we will heat it up. And this you do before you sterilize it usually, because agar has a useful property. It liquefies at 100 degrees Celsius. We would then usually take that melted agar, sterilize it in an autoclave, and then do whatever we have to do with it. We can use it to pour Petri plates, for example. That is a common way of using it. We pour it into a Petri dish. We can pour it into tubes, and we can incubate those on a slant so that they solidify with a nice big elliptical surface. Or we can let our tubes solidify while they are upright like that. And that provides us what we call a deep. We often use a deep for storing bacteria because we can stab deep into the agar, inoculate deep into the agar. So how we use the agar depends on the setting, depends on what we pour it into and how we keep it. Here's the other very important characteristic of agar. I said it liquefies at 100 degrees Celsius, but it only solidifies at around 40 degrees Celsius. At around 40 degrees Celsius, you can pour your plates, whatever, at 40 degrees Celsius. Not only is that useful for the microbiologist because you take it out of the autoclave and you leave it there, and you can leave it there for ages before you pour your plates, but you can mix bacteria with it, as you'll see in a while, because bacteria will survive quite well at 40 degrees Celsius. You can inoculate it with bacteria and then pour it. And you'll see some of the reasons that we might want to do that, we'll see in a while. Just remember important characteristics of agar. Liquefies at 100 degrees Celsius, boiling point, only solidifies at 40 degrees Celsius. Okay, just examples. Here's a chemically defined medium. This you could grow E. coli and many other things on. Look what it has. This is why it's a defined medium. First of all, it has glucose in it. We know exactly how much glucose. We measure it. Ammonium phosphate, sodium chloride, magnesium sulfate, potassium phosphate, and water. Each of these components is carefully measured. We know its exact weight. We know its exact molarity and everything else. When you look at this, the reason E. coli is able to grow on this is because it can manufacture many of its own, as long as it has a good source of carbon and energy, and then it's got a source of nitrogen, for example, and then some other things, magnesium, potassium, a phosphate source, etc. Here, it can manufacture everything it needs. It can manufacture its own amino acids. It can manufacture its own growth factors. All of those things, it can manufacture all of it. Bacteria very often are extremely good at working like that. They are completely independent of rich nutrient sources if they are not present. So that's a defined medium. By the way, E. coli grow well on a defined medium. E. coli grows equally well on a complex medium. And that is how we usually grow it. So this is a chemically defined medium. This is a standard laboratory level complex medium. And this is a sort of medium you would encounter in a microbiology lab to grow most of the bacteria that you would see. So here's what we mean by being complex. Here's the peptone. It's digested protein. We cannot say what the exact composition is. We can't say exactly how much of each amino acid is present. We just know it's in abundance. We can't even say how much nitrogen, carbon there is there. We just know it's in abundance, more than they need. Beef extract. Beef extract is basically boiled down soup of beef, and it contains beef. It contains growth factors and various other things, which are basically just useful to the bacteria. Then the sodium chloride is basically to establish the tonicity of the medium. It's to keep the medium isotonic. And quite honestly, it's not always included. Then there's water that makes it a nutrient broth, and we solidify it here with agar. And 15 grams of agar per liter is the standard usage of normal agar, of normal laboratory grade agar. So this is a very, very typical. You can take this recipe and make this very easily and use it for growing most of the bacteria that you would see in a clinical lab. There are some other specialized media, though. We encountered one of them already when I told you, when we looked at the use of oxygen in those tubes. I told you that very often we will do that in a specialized medium where we include something which binds up most of the oxygen in the medium. That is called a reducing medium. And the most commonly we use this substance, sodium thioglycolate. And sodium thioglycolate just binds up the oxygen present in the medium, and it is no longer available to organisms. It does allow oxygen to percolate into the medium at the very surface, so you set up a gradient. When you use sodium thioglycolate medium, very often what you will do is just before you inoculate it, you heat it up to drive off all the oxygen. You let it cool and then you inoculate. Then an oxygen gradient will establish from the surface down if you leave that surface open. But the bulk of the medium will be free of oxygen, and it's a very good way of growing obligate anaerobes or observing facultative anaerobic growth. So that's a reducing medium. It's a medium in which oxygen is depleted. There are other ways. Often anaerobic bacteria can be very difficult to grow, particularly if they are fastidious and if they are obligate anaerobes. And if that is the case, depending on the bacterium, very often if you were trying to grow them, if you were trying to culture anaerobic bacteria, what you often do is you'll actually get bacterial spores, inoculate media with bacterial spores, and then put them into an anaerobic environment, and then they will grow. The bacterial spores will not germinate in the presence of oxygen, but they will germinate if there's no oxygen present. This is not particularly difficult to do. This is a typical anaerobe chamber. It's simply a sealed vessel in which you put a special combination of chemicals. You buy them and put them in. You open the little envelope, and what happens is that they actually convert all the oxygen, and they convert it into various things other than free oxygen, and they usually release carbon dioxide and hydrogen. Their working is a really complicated thing, but they work pretty well. And you usually include in that an indicator so that you can see that it is truly anaerobic inside, but there's absolutely no oxygen. And these are the petri dishes probably inoculated with bacterial spores, either something like Clostridium or Bacillus or whatever it is, and they would then grow happily because this is anaerobic. So this is an anaerobe. This is an anaerobe jar, it is called. On the other hand, there are anaerobic bacteria like some of the Clostridiums, which are so fastidious, so absolutely averse to oxygen, that even the slightest trace of oxygen in their environment at all renders them unable to grow. They simply shut down and they will not grow if there's a molecule of oxygen present. And particularly if you're having to grow a lot of them, that can be very difficult. So in an anaerobe lab, they will often have these chambers. And they're anaerobe chambers, and they have an airlock. Everything going in and coming out passes through this airlock, which is evacuated completely before you move anything. There are arm ports that you put. They actually have gloves on the other side usually, and you stick your arms through and you work inside here, so that this is kept completely free of oxygen, and it is perfused usually with a mixture of gases, nitrogen, sometimes with a bit of carbon dioxide, that sort of thing, and it is continually evacuated with those gases to ensure that there is absolutely no oxygen, and there may be an oxygen trap somewhere just in case there's a spare molecule of oxygen somewhere there. You have chemicals there to grab onto that oxygen to ensure that it is completely anaerobic. Now, there's another group of bacteria, and they're quite interesting because we do encounter them clinically and in the clinical laboratory. And these are called the capnophiles. The capnophiles are interesting. They are aerobic. That is, they grow in the presence of oxygen, but they like to have high concentrations of carbon dioxide. And there are different ways of doing it. Nowadays, you can buy a packet that you open, and it produces enough carbon dioxide for them. And this is a capnophile jar. You could actually do this with an envelope of this capnophile mixture. But the classic standard way of doing it is to run what is called a candle jar. So here are media inoculated with bacteria that we know are capnophilic. And then we grow them, we put a candle in there. And the candle burns down and eventually goes out. And that's a perfect mixture for them. That's a perfect amount of carbon dioxide for them. And this is the classic way, and you will still see it done in many labs, especially if they do a lot of these and they don't want to waste money on capnophile envelopes. It works perfectly well as a capnophile jar, it is called, or a candle jar. Okay, so let's think. We've heard now about like the defined media and the complex media, but we can design our media to do various things for us. And remember that in a clinical laboratory, very often you are going to start out dealing with samples which are a mixture of bacteria. You may want to purify bacteria from that mixture. So you may want, first of all, to somehow select. You may want to use your media to somehow select things that you might be interested in. And to do so, we use selective media. These are media, and usually solid media, which have components in them which are going to suppress unwanted microbes, and they will encourage the ones that we're interested in, increasing our chances of being able to sample the ones that are of particular interest. So they've got, usually selected media contain something in them which inhibits growth of the ones that we are not interested in. And typically, for example, there are media which will suppress the growth of gram-negative bacteria, say. And those will encourage the growth of gram-positives, the other way around too. There are media which will suppress gram-positives and allow gram-negatives to grow. Now, those would be selective. Then we can also make media which are differential. And that allows us to distinguish between different bacteria growing on the same plate by emphasizing colony characteristics. Now, bacteria growing on solid media very frequently have particular colony characteristics which we can recognize. So, for instance, they may have particular shape, they may have particular color, they may have particular margins, where we learn, okay, I know what E. coli looks like growing on such and such a medium. But differential medium very often allow us to distinguish from an unknown mixture. They start to allow us to distinguish between functional differences between bacteria growing on that plate. Very often, media combine both of these characteristics. They are both selective. They suppress growth of something. They allow growth of other things. And then they are differential. Those things which do grow, we can distinguish between them because they show different characteristics on the medium. So we'll see some examples. So this is a differential medium. This is not selective at all. In fact, it's a good generalist medium. This is called blood agar. Blood agar is it's a nutrient broth solidified with agar, but included in the agar, in the mixture rather, is sheep's blood usually, and it's entire blood. These are entire red blood corpuscles. The corpuscles are, if you were to look at them under the microscope, they would look pretty normal, and that's what gives it its red color. But the blood agar is a differential medium. Many things will grow on it, but some things that grow on it are actually able to interact with the red blood corpuscles, and they do it in different ways, and that's what makes it differential. You can see here, this is a bacterial colony there. That's a bacterial colony there. Surrounding these ones, there's a clear halo, and that we call that clearing we call hemolysis. And there are different characteristics of hemolysis, which I'll show you. They get a Greek letter. Let's have a look at this one first, even though it's the second letter. This is beta hemolysis. There's the beta for you, okay? And beta hemolysis completely clears the blood agar, because what happens is the bacteria growing here are able to release enzymes which lyse the red blood corpuscles and then completely break down the hemoglobin. And it ends up, you just get the native color of the medium with no blood coloration at all. So that's beta hemolysis. So therefore this here would be beta hemolysis. This one here, you may not be able to see anything at the moment, but you'll be able to see it once I describe. This is alpha hemolysis. Easiest way to see it, look in the middle here. Can you see how this and around the edges here has acquired a green tinge to it? That is typical alpha hemolysis. Alpha hemolysis, they do not break down the red blood corpuscles. They do affect the hemoglobin. They partially break it down, but they don't break it down completely. And one of the breakdown products from that process has a green color to it, which gives alpha hemolysis that typical coloration. The last one is called gamma, and it's nothing. Gamma hemolytic bacteria, they're not able to hemolyze at all. Only alpha and beta are. So that's purely differential. It's not selective at all. Here is another differential medium. This is one that you would or may encounter in lab. This is called mannitol salts agar. And it is, as its name suggests, it contains a sugar called mannitol, and it contains salts, a lot of salts. Now remember back, we said one of the important environmental things that we have to consider is the osmotic environment. Mannitol salts agar has a high concentration of salt, about 5%. Not many bacteria can grow under that kind of regime. They just completely plasmalize and die. But there are certain bacteria which can survive. And this is especially true of this group of bacteria, the Staphylococcus. Here we have Staphylococcus. In this experiment, they have not inoculated here. This is uninoculated. So the Staphylococci grow perfectly well on mannitol salts agar. It is actually selective, strongly selective for Staphylococci. Why? Well, have a look at the name here, and you'll have a clue. Staphylococcus epidermidis grows on the skin. So does Staphylococcus aureus. Staphylococcus grows commonly on the skin, and it is subject to our sweat. And it is adapted to a high salt environment. That is why it grows very well on the salt medium. but you notice something else. And that is that this Staphylococcus epidermidis, it is red. This Staphylococcus aureus is yellow. This mannitol salts agar is strongly differential because it allows us to tell the difference between the two staphylococci. It is selective because not many other bacteria will grow on it, but it is differential because Staphylococcus aureus here ferments mannitol, and it produces an acid, and it changes the color to yellow. Staphylococcus epidermidis does not ferment mannitol, and so you can use mannitol, but doesn't ferment it. The Staphylococcus epidermidis does not turn this yellow. We can distinguish between these two species of Staphylococcus by their reaction to the mannitol. If we were to inoculate in some other bacterium, E. coli or something here, it wouldn't grow at all. And so that would show us that this medium, mannitol salts, is strongly selective for Staphylococcus, but it also differential. So it has both qualities. Here's another one, which is also selective and differential. This is a very, very famous medium called McConkey's. And here it is here. And McConkey's is used especially to select for a particular group of bacteria which are common in our gut, the Enterobacteriaceae. McConkey's has two components to it, which are inhibitory. And these are bile salts and crystal violet. And these inhibit the growth, first of all, of gram-positive bacteria. They stop that gram-positive bacteria from growing. And we're going to see that there's also a differential property to this medium as well. So first of all, let's just grow our bacteria here, our experimental bacteria, on ordinary agar. This is nutrient agar. So here's E. coli, and E. coli grows perfectly well. Here's Staphylococcus aureus. Staphylococcus aureus is a gram-positive. It's a gram-positive coccus. Now we're going to take the same two, and we'll grow them on McConkey's. Here's the E. coli, grows perfectly well. Here's Staphylococcus, completely inhibited. It is inhibited by the bile salts, and it's inhibited by the crystal violet as well. The bile salts is interesting. Why do you think E. coli is tolerant of bile salts? Bile salts are produced by our liver, and they're liberated into the gut to emulsify fat during digestion. So our gut contents actually are saturated with bile and with bile salts. So bacteria which normally grow in the gut will be resistant to bile salts. And E. coli is typical of that. There, E. coli is typical of that. And so this is strongly selective for this group of bacteria, the Enterobacteriaceae. But McConkey's is also differential because it contains a sugar, lactose. And when lactose is fermented, it turns the medium yellow. This E. coli here is unable to ferment that lactose in this medium, and it remains this dark color. Salmonella enterica is able to ferment, and it turns the medium yellow. So there's a differential feature to McConkey's. If we grow two Enterobacteriaceae on it, we may be able to differentiate between them. If we grow two bacteria of different gram reactions, one will grow, the other will not. So McConkey's is both selective and differential. And that is very common with these kinds of media. We get a lot of information from these media, and we frequently use them in the clinical setting. They are very, very frequently used. They quickly give us some signal characteristics of the bacteria that we are dealing with. Okay, so this is just a summary for you. This is, we saw chemically defined media, where we know exactly. Complex media, with our generalist kind of media, which most of our chemoheterotrophs will grow on perfectly happily. Reducing media with deliberately created media that have low oxygen content, and that is for growing of anaerobes rather. Selective media, we suppress unwanted microbes and encourage the ones that we're interested in. And then differential media, those which produce colonies where we can easily see differences in metabolic reaction. Okay, so again, this bit here, I'm including because I know that your laboratory experience is going to be somewhat limited. You may well do these exercises, but it will help you to actually understand something about what you're doing and the reasons why you're doing certain exercises in the lab. Always in a microbial laboratory, we are always concerned with knowing exactly what we're working with. We need to know that the bacterium that we are growing is actually the one that we intend to grow. And for that reason, we usually try to get our bacteria to a status of a pure culture. Now, we may have derived our culture from a known culture and be wanting to maintain it, carry on growing that culture. On the other hand, we may be looking at a mixture of bacteria and trying to pick out the component bacteria, then grow them sufficiently in a pure state that we can analyze them and identify them. In either case, we're going to require pure cultures which contain only one species, or sometimes higher than species, one strain, one bacterium with one particular characteristic. So just some terminology. A colony is the growth which we observe on a plate. Let me just go back here. Okay, well, we can use this one for the moment. That's a colony there, growing on a plate. And ideally, in a microbial setting, when we're culturing, what we want to see is we want to see isolated colonies like this, because the implication is that that colony there has grown from one cell, dividing rapidly. Each of these has grown from an individual cell. So when I pick a colony like this, a just visible colony on a plate contains about 1 billion cells. But all of those cells, that billion cells, have been derived from one single cell. They all have exactly the same genetic characteristics. That enables me to establish a pure culture. I can take one colony and I can grow it up and propagate it on and on and on, and know that it will always have the identical genetic properties. So that is the ideal. We always try to work from individual colonies like that, where we can assume that they have grown from a single cell, or sometimes if you're using anaerobes or fungus, a single spore. We can't always tell that we can't always be strictly sure that that is the case. So sometimes, just for purist's sake, we would refer to a colony as a colony forming unit. As we'd say, it has been formed from a colony forming unit. It may be 2 cells. We can't be absolutely sure. We hope the ideal is that it grew from one, but purists would say you should refer to it as a colony forming unit. In the laboratory, the quickest and the most frequent way to move from culture, especially from liquid culture, onto media and ensure that you end up with single colonies, is to use some method of diluting the bacteria enough on the surface of a plate that you end up with single cells separated from one another so that each will form a colony. This is the way you would do it in a lab. This is what you would learn to do in lab. The most common way, the quickest and the easiest, is to do what is called a streak plate. So what we're going to do, we're going to, first of all, we'll have a source. In this case, it's another solid agar plate. And it could easily also be from a liquid broth culture. And we're going to take a sample. When we take the sample, we've got a gazillion bacteria. And we're going to use what is called a microbiological loop. It's a wire with a little loop at the end. The very first thing you do is you have to sterilize that loop. You pass it through a flame so that any living bacteria, any spores are burned off. You cool the loop and you take a sample. In that sample, in that inoculum that you have taken, you have billions and billions of cells. We have to somehow separate those cells. We have to end up in a situation where we have cells separated on the surface so that each cell grows a pure colony. This is how you do it. You take that sample and you spread it like this in a wavy loop, back and forth and back and forth and back and forth. Now you take your loop and you flame it. You kill all the bacteria on it. And now you do another wavy loop. Here it is this way, that way. Now let's just stop here and think what we've done, okay? We picked an inoculum with many billions of cells. We spread them out. What did we do when we spread them out? We diluted them on the surface. There's still a lot. There's still an awful lot of bacteria. But there's less here than there was sitting on the end of your loop. So now you flame your loop, and now you do another spread like this. What have you done? You've only picked up that many bacteria, and you're spreading them out over this whole area. Again, you've diluted over the surface. Now you do it again. You flame your loop, and now you spread again. And what have you done? You've taken only that many bacteria. Remember, this is much, much less than there. You've taken that many bacteria, you spread them out here. Now, we usually just do a single, you flame the loop again, and now you just do a single swipe. You're only taking a very few bacteria, and you're spreading them out all along the streak, like this. As you go, you are diluting them. You're taking individual cells and spreading them far away from one another, and this is what you end up with. Here's your first streak, thick with bacteria, right? There's your second, but all you've done is you've picked up a few far fewer than there are in the whole thing. You picked up a few there and you spread them over there. Here, you've done the same thing. You spread all the way here, and now you've spread here, and here at the end of your streaking, you end up with beautifully separated colonies. And you can be pretty sure that each of these colonies arose from one single bacterium. So all that you're doing, the streak plate, is you are actually diluting. You're diluting as you go along. A lot, fewer, very few, all spread out. And you end up with your single colonies. So let's just talk very quickly, and I'll just do this section, and we'll finish for today, and possibly repeat some of this next time as well. Let's talk about how it is that bacteria actually do divide. And bacteria divide by binary fission. They grow to a certain size, and there are cellular signals which are sent to the cell to instruct them to divide. They have, bacteria have a single large circular chromosome. The chromosome is entirely duplicated. Along with that, by the way, if there are plasmids and other things, those are also duplicated. And both the cytoplasm and the genetic material is apportioned equally to the other side of the cell. And then the cell pinches off and a new cell wall is developed to produce the two daughter cells, which then continue to grow and go through the same process. In bacteria, this can be an extremely rapid process, especially if they're in an appropriate medium and if they are at an appropriate temperature. In E. coli, at the height of its growth, this can be accomplished in between 12 and 20 minutes. They can divide like this so what we need to realize, in that time, in that period of time, let's give them, say, 20 minutes, they double their population. Because under ideal conditions, every cell divides. And so every 20 minutes, you double your population. You start off 1, you go to 2, to 4, to 8, 16, 30. It is an extremely rapid process to develop a very, very high population of bacteria from relatively few bacteria to start off with. And when we watch their growth in culture, we see that there are particular characteristics. Okay, let's just, if I can just find this. Oh, there we go. No, sorry. I've got a thing which is right in the way of the little arrow that I have to do to, there you are, there we are. Okay, all right, so let's leave it there for today. Before you go, please unmute if you have any questions.
I have one question.
Yes.
So the cross wall, that's the septum that they were talking about in the video, right?
The cross wall is actually the cell wall. The septum is where the cell membrane develops, and then the cross wall is the actual cell wall that then develops outside of the cell membrane. That's when they separate completely.
Okay, thank you.
Anyone else?
Yes, Professor, I just had a question about the culture technique with the capnophiles. So with the jar and the candle, I see in the image, there's a stack of Petri plates.
Yes.
And then there's tubes with media, liquid media. So I was just wondering, are the bacteria that you're encouraging to grow in the high CO2 in the liquid media, or they're
both? It can be either.
Okay.
Yeah, it could be either. Yeah, it doesn't matter. If you've got these tubes with the liquid media, what you do is you usually leave the caps very loose. You want to keep the caps on to stop any possible contamination, but you leave the caps very loose so the CO2 percolates in. But it's just to show you can use this technique with liquid media as well as with solid media.
Okay, so the Petri dish, the Petri plates would also be like a little bit askew with the lids so that there could be...
Correct. Petri dishes allow a lot of percolation of air, of gases. The gases get in and out very easily. It's very loose. The Petri dish is very loose in the lid.
Great, thank you.
Sure. Anyone else? Okay, everybody, if you don't have questions, thanks very much for coming in for the live class. We had quite a number of people coming in, which I'm very pleased about. The class is recorded and will be posted as normal. And if you've got no more questions, then I'll bid you goodbye. Wish you a happy weekend.
Professor, when is the next exam?
I beg your pardon? The next exam, did you say? I think that the next exam is the 21st. Am I right?
This month?
Yes.
21st? Okay.
21st. I think it's the 21st. I will send, as usual, I'll send reminders to people that and it's an order, although it says midterm, it's not a comprehensive of the whole semester. Okay. It's a normal test. So it'll be stuff that has not been examined yet.
Okay.
Will not include what was on the first test.
Thank you. So it's gonna be on chapter 6, 7, and what chapter are the exam gonna be?
Yeah, I can't think about the moment. Yeah, it's something like that. I'll tell you what. I'll tomorrow when I do your assignment and everything, I'll send an email reminding everybody and telling you exactly what to cover.
Thank you.
All right.
Have a good day. Bye bye.
Hello. Okay.
Yeah, I said thank you.
Anyone else? All right then. Okay, everybody. Bye-bye, and see you again soon.
Okay, thank you, Professor.
Thank you.
That's a pleasure. It's nice to hear your voices. To know that you're out there.
Maybe we should do like a in-between session while you're lecturing. Maybe we can jump in and like talk sometimes, I guess. Maybe that will work.
Well, yeah, I do periodically break it up in the middle of the lecture. It's just that I find it difficult to gather everybody again afterwards. I observe that when I do that, if I have a break, I see the numbers drop immediately because people just don't come back. I say, take a break or whatever. But maybe I can stop and just say, hello, everybody, are you out there? What's your name, etc., etc.
Okay, I understand.
All right. Okay.
Thank you again, Professor.
That's a pleasure. Nice to hear you. Yeah. Bye.
Have a good night.